So what I thought I'd do today is give you just a brief overview of the Allan Institute for Brain Science. You guys are going to be there this evening for dinner, and we're going to do a tour, but at least a little bit of an overview. And then I'll jump into the Allan Human Brain Atlas project, just kind of fly through that a little bit. And then Christoph Cook, in a couple of talks, will talk more about some of the new stuff we're up to. So history of the Allan Institute for Brain Science, one of these fortunate times when Paul Allan is actually in the room as I'm talking and introducing this. History goes back a little over a decade with some interesting planning meetings. And really over the last decade, we've been shaping this organization to create and act as a fairly different organization in the research space, where we create products that are large data sets with tools and information that are readily available to the broader science community. We are organized and structured much more like a biotech company than a traditional research organization. We're very project-focused, multidisciplinary teams of a lot of different disciplines, again, all coming together towards these common goals. Right now we have about 200 people on board. There are 43 of those are roughly are PhDs. And as you'll see this evening, we're located in the Fremont area of Seattle, which is just basically down the waterway here, about 2 or 3 miles. What we are not is a traditional PI-driven research organization, and we are not an extramural funding agency. I always like to highlight that. So when the Institute was formed, there was a lot of discussion around what should the Institute pursue. And there was a broader mission of saying we really want to understand how the human brain works. I think we can appreciate that in this context. And so there's, of course, many, many different places in which you could start. And we've heard a lot, I think, early today around these areas here, which are very, very hard, as we've sort of, I think, had some very good talks and comments relating to that. The decision was made, I think, from a variety of different science advisors, sort of converging on the idea that we would like to start at this end of the spectrum. Let's try some reductionist approaches to really start to understand the component parts and build up from there. There's a lot going on here, and we'd like to systematically start to build these resources from here to move in this direction. And what we've done over the course of the last decade is start to create resources by looking at where genes are turned on in the brain. We started with the adult mouse brain. We've subsequently generated resources on a variety of different aspects of brains and species. So we've looked at the developing mouse brain. We've looked at the mouse spinal cord. We've looked at the human brain. I'm going to focus on the human brain today, and the non-human primate brain as well, both in development and in adult. Moving forward, we've started exploring. We have a project right now that I'm not going to talk at all about today anymore, is looking at the connections, the wiring of the mouse brain at the individual wiring level. And moving forward, Christoph will talk more about our efforts in computation, dynamics, circuits, behavior. So the topic of our Seattle Brain Salon is molecules to mind. We've been talking a lot about the mind. Let's talk about the molecules. This is a fly-through. We've been focused again at the gene level, 25,000 genes in the genome, and then this is a human brain slab, just showing a little bit of the perspective of the approximately 86 billion neurons in the brain. And so we've got layers and layers of complex systems upon complex systems from which to look at. So it's really been our approach to try to bring together this information, starting at the gene level, seeing what genes are turned on in areas of the brain, and then providing useful free tools on the web for people to analyze and look at the data. This is one tool that we've created. It's called Brain Explorer, that's showing brain that we've extracted information from. The little colored dots there were a single gene that's been turned on as we've measured it in each brain. We measured about 1,000 times. I'm going to tell you how we do that. Okay. So, but before I do that, I mentioned the web-based tools, just a little bit about our impact and the kinds of things that we do. So we have a web portal I just highlighted all the different projects that we do there. It's brain-map.org. Roughly 45 to 50,000 unique users come in and mine and use the data on a given month. We know that these are people from all walks of life in the research community, pharma companies, biotech companies, academic research institutions, and studying every aspect of brain biology. Is a nice part about doing these broad-based resources on something like the brain is that people studying almost any aspect of brain biology can find some value in the resources that we've created. So since inception, just a little institute in numbers, we've generated data on over 3.2 million tissue sections, almost 1 million microscope slides. That's easily a petabyte of image data. That used to be a big deal. Petabytes aren't such a big deal anymore, but at the time we started, it was a big deal. We provide engineered mice. We do a lot of transgenic work. It's just in support of our cell types and our connections projects. And lots of gene expression measurements. And all this is free and available to scientists around the world. So let me talk to you about the Human Brain Atlas. I just said deconstructing the brain in several hundred easy steps. So everything we do, we try to do in a very industrialized, structure-heavy, standard operating procedure-heavy way so that we have the highest quality data that we can possibly generate. And this is a broad overview. We take a human brain, we collect data on it. This is a dead brain. It's post-mortem. We do collect, in a post-mortem state, diffusion tensor imaging and MR. We then take the brain out, we slab it, freeze it, do histology on it, stain for individual cell markers. That gives us an information base to give to a team of annotators, which then mark it up. We do anatomic sampling in the lab. We extract RNA. We put it on a microarray, and we get a digital readout. We get a quantitative readout of genes that are turned on in that particular part of the brain. For the first 2 brains that we process, we roughly partition this brain, each adult human brain, into about 1,000 parts. And as I mentioned, then we extract all that information, put it all on the web. So here it is in a little more detail. A lot of people are fascinated by the human brain collection process. Obviously, for those of you who work with the living brain, it's easy when someone comes in and then they leave. People come into the medical examiner's office. We have relationships with medical examiner's offices in both coasts. This one is actually the state of Maryland. The entire state of Maryland is in one medical examiner's office. So there's about 4,500 cases a year. We screen very hard for normal. We had a good conversation at lunch about normal. Because they're so infrequent, roughly we're able to obtain anywhere from about 5 to 10 normal human brains a year using this method. Ages 20 to 60, no history of psychiatric disease. We do a full tox workup to make sure that there's no serious drugs on board other than caffeine or nicotine. This is an example of the MR and the DTI that we take in a post-mortem state. In Maryland, we're fortunate in this. There's a clinical radiology core right next to the brain bank, which is right next to the medical examiner's office, so they can actually wheel the body in and do the MR there without having to remove the brain. The brain is then processed, it's removed from the skull, it's slabbed, it's frozen, and it's sent to Seattle. And you'll get a little demonstration of this evening. But this is a frozen slab. It's basically put into a large whole body cryostat, and we develop methods for being able to cut this fresh tissue. It's not fixed at this point because we need to preserve the RNA. Our criteria for selection of brains is also that we need to have this frozen state no less than 24 hours after time of death. So you can imagine all the logistics around obtaining consent for obtaining the brain, and then getting the team mobilized to collect the MR, the DTI, and then remove the brain and get it frozen within 24 hours. So there's a lot of pieces that have to come together to make that happen. Then anatomy. So we've seen a lot of free surfer representations. Obviously, we've collected the MR, so we apply free surfer to that. But we also collect Nissl stains and have a team of anatomists who are expert at recognizing structures go in and annotate the structures. And they hand that information over to a group that does partitioning. If it's a cortical area, I'll show you that's a scalpel dissection for obtaining cortical areas. If it's subcortical, We do laser capture. I used to have a movie in here, but basically, for those of you who don't know laser capture, you see a picture of a scan on a screen. You scribe where you want the laser to cut. It cuts. The sample falls within a tube, and you extract RNA from it. So this is our macro dissection workflow. As I mentioned, areas are described by anatomists, cut out, and then put onto a microarray for a readout. Same thing happens for laser capture, microdissection. This just shows part of the hippocampus that's been collected. One of the things that we pride ourselves on doing is allowing everyone to see every step of this process. There are lots of data sets out there which are a microarray data set that's a table, an Excel spreadsheet, and you don't really get any kind of additional information about it. So all of this information of just seeing exactly what we extracted. If you're curious about CA4, you're wondering if we might have gotten it wrong, you can see exactly what we took in terms of matching that up. And we have all kinds of back-end lab information management tracking to make sure that indeed the sample that we say it is here is the one that you're looking at in that data. Okay. And again, this gives you microarrays. So, for example, something like the hippocampus, I think I actually tried to write the exact number of microarrays here, but this ended up being, I think, about 63 samples just for this part of the hippocampus. This is put onto a microarray. For those of you who are not familiar with microarrays, this is basically a small piece of glass on which there's oligonucleotides. These are 60mers. This is a technology developed by Agilent. And there's about 60,000 of these elements on there. So we actually can, for many genes, have 2 oligonucleotides which are reporting the activity in that sample. We were able to also tile through genes, and so we were able to assess quality and do a number of things with this particular data. And then what this does, when you label up a sample with RNA, put it on there, you get a quantitative readout, and then we provide all of that information out. And as I mentioned before, we have web-based tools for that. So what can you learn from this kind of information? And I think it's really important to note, I mean, really for the first time ever, we've got a resource here for the entire adult human brain where you can look at the activity of any gene in the genome. Okay? It's very powerful. And in this case, for example, here's something where we've gone ahead, and this is all the structures and samples here. Remember, this is roughly about 1,000 samples per brain. This is just looking at the first 2 brains we've collected. And it's color-coded by the different areas. And then we decided to let's just look at the monoamine pathway. Let's look at all the genes that are involved in dopamine synthesis and processing and reception, and let's look at them all at once and see what they look like. And you get a snapshot of this kind of stuff. And of course, all that data is there for deeper mining. If you'd like to, for example, say, I'm particularly interested in the dopamine 1 receptor, but I'd like to see everything else that sort of comes along for the ride with that. You can just do a correlational search, and out pops things that look very similar to it in terms of its pattern. Mining a little deeper in this, we were very interested to see, because we sampled the cortex quite broadly, we pulled about 600 samples across various cortical areas. We wanted to see in sort of an agnostic way, can you just look at the data and have it organized in some way? It's sort of, this is basically considered a principal component analysis, where the gene expression is actually recapitulating the relationships as you might see them anatomically. So there's clearly lots of information in here about the areas that we're extracting that are telling us a lot of information about the underlying biochemistry related to these functional areas. Okay. So we had another study that we did where we looked at 1,000 genes using in situ hybridization. In situ hybridization is a different technology which allows us to look gene by gene, get it at the cellular level, but again, it's just gene by gene. So it's a different kind of technology. It's more qualitative technology. But we were very interested in looking at the neocortex and seeing similarities and differences across individuals and different cortical areas. And so to the point of that last slide, which showed these areas, indeed, when we looked at those 1,000 genes, roughly about 15% of them showed differences across different areas of the cortex. Again, as we're thinking about cortical maps and thinking about activities and thinking about trying to draw in individual areas and start to link them to underlying biochemistry, this is a very promising result, and that there's definitely differences in cortical areas, and there's underlying biochemistry behind it. Okay, so these are just some examples of that. The next thing we were very interested in doing, because our N is exceptionally small in these kinds of studies, the amount of this millions of dollars of investment in each brain that we collect in terms of the data. And so at this point, we now have data collected for 3 whole brains. This analysis is just showing the first 2. And very encouragingly, if we take a reasonable cutoff of 150 structures and the sort of top 500 high-confidence genes, we see a lot of similarity between brain 1 and brain 2. And this is very encouraging. Given all the variability across humans, I guess it didn't necessarily have to be this way, and so we're encouraged, given all the different steps taken, and keep in mind these are 2 unrelated individuals. I found this correlation to be quite remarkable. And again, back to that study that we did with in situ hybridization, we had evidence for this looking at 1,000 genes in much more detail, where we see roughly about 5% of the genes vary across individuals. And here's some examples of things like relin and DISC1. Both are for those who are developmental or disease-focused folks, you'll recognize these genes, but they're basically different in individuals that we looked at. We're also adding to this what we're calling a phase 2. So we're now we're taking a hemisphere and processing it using that microarray approach. And the other hemisphere we're blocking out, and we're doing in situ hybridization. And we're focusing on excitatory and inhibitory gene systems. So we want to get the cellular resolution data. It's hard to get because it's gene by gene, so we're focusing on just a few high-value genes and running through and generating that data as well. And it looks great, so that you can go through areas like the hippocampus and see things like GABA theta subunit in the dentate gyrus. So one other thing that I didn't put in here, Christoph mentioned, that I probably should at least highlight briefly, and I'll talk a little bit more about this evening when you're at the Institute, is we have a whole other project that's sponsored by the NIH on the developing human brain. That's a combination of gene expression and in situ hybridization as well. And we've been doing work on the developing non-human primate, the rhesus macaque monkey. And again, all of that data is available and free for people to access and use. So I'm actually running just about on time, so I wanted to, given that I think I'm the only person really talking about genes in the session on genetics, I don't know, I'm not guaranteeing that given the gentlemen that are coming after me, but I kind of think it might be the case. What's coming down the pike in terms of, not from us per se, but the technologies and the data streams. And so I highlight 3 very recent examples. There were 3 letters to Nature in just a couple months ago where they've gone in and they've looked at sporadic cases of autism. And autism is one of these things where they're looking very hard for the gene or the genes. And when they go through, and they can do this now, you can sequence for very cheap. So they're going in and they're sequencing the exomes of all these individuals that are part of these larger collections. And they're finding just this mind-blowing result, which is there's probably 200 different genes that seem like they're implicated in this. And you can find the deletion in these individuals. And so imagine now that not only you're going to do this with autism, but many, many things. Sequencing is becoming so cheap. This information is going to become readily available and start linking to all kinds of things. We've had some good conversations at lunch around starting to link to a lot of different human phenotypes that aren't just necessarily the heavy, big genetic diseases, but just about anything that one can measure. And so I think this is a very exciting time to be starting to think about genes as they relate to in phenotype, right? So you have the molecules, and you have the end phenotype of the mind, and there are important things to learn The other thing I want to highlight, and this is something that we have a new program that we're launching at the Institute to start to do more of this, and Ricardo Dolmetsch, who's one of the authors of this study, is announced that he's joining us from Stanford, is that you can take a skin pinch from any one of you in the room, and you can get those cells to de-differentiate to a native state, and then you can get them to redifferentiate down a neuronal state, and you can make neurons in a dish. And more sophisticated approaches are being developed to make different kinds of neurons in a dish, and maybe collections of neurons in a dish. Some of the examples, here's one on Alzheimer's. Ricardo's first example was taking cardiac cells. There's a cardiac defect in people with Timothy syndrome, and they can actually recapitulate this in the dish. So I think the wave of the future, things that are coming, are this ability to do tests of humans with specific genetic conditions that we know are going to relate to brain function, and be able to start to test emergent properties out of those. And I can't imagine that there aren't going to be great discoveries, especially as they relate to disease, but I think more fundamentally to just how these networks of genes ultimately relate to the complicated network of the mind that we've been hearing so much about earlier today. So let me just do a broad thanks. Again, Paul is right here. I can thank him directly. A huge number of people. This is actually an older slide. There's probably another 100 people associated with the Institute at this point. And not only the people that work at the Institute, but one of the things we pride ourselves on is we have lots of advisors and lots of collaborators that help us in a variety of ways create the kinds of things that we do. And I always point to the website because they're better acknowledged in a more full sense there. I usually have 20 slides if I actually try to put it on slides. So with that, I'll conclude, and thank you.
Thank you.